Good afternoon, and thank you for joining us for the virtual March of Dimes High School Science Week. I'm Salk President Rusty Gage. For the past 30 years, the Salk Institute's had the pleasure of hosting this March of Dimes High School Week, a week where we invite high school students to tour our labs and meet with Salk scientists to show students a glimpse of what a day in the life of the scientist really looks like. While we're not able to invite students to our labs in person this year, we're thrilled to host you in a virtual lab tour. This week, we will have opportunity to interact with scientists from a wide variety of Salk labs, including my own, which will be featured on Thursday, as well as hear from a few of our esteemed faculty members. And now it's my pleasure to introduce our opening speaker, Dr. Jeff Wall. Dr. Jeff Wall is a professor in the Gene Expression Laboratory, where his work focuses on better understanding cancer. Particularly, he focuses on breast cancer and pancreatic cancer in order to develop improved and more individualized therapies. And now, to tell you more about the Salk Institute, about his lab work, and about the work that he's doing for the rest of us, world-recognized leader in cancer research, Professor Jeff Wall.
Welcome, students. Thanks so much for joining us at High School Science Week at Salk. This is a great pleasure for me to present because I was actually, as I recall, the leadoff speaker at the first High School Science Day at Salk 31 years ago. Rusty, thanks so much for your very kind introduction. And today I'd like to talk about four things. First, I want to give you a brief introduction to the Salk because you're going to be visiting this week. I'm going to tell you a little bit about my background, and I hope to impress upon you that any person who's inquisitive, who's interested in the world around you, regardless of your background, can become a scientist. But to become a scientist, first you have to understand what science is, and I'll tell you what that is from my perspective. And then I'll tell you what my passion is, what I've been doing since I've been at Salk. And basically, it's been doing cancer research, as Rusty Gage said. And the objective is to get foundational knowledge. I don't call it basic knowledge or basic science, but rather foundational, setting that great underpinning upon which subsequent work can be done, because this generates the knowledge that's needed to improve people's lives. So the Salk Institute, started by Jonas Salk after he developed the polio vaccine. The March of Dimes gave him funding to start an institute that was a collaboration. The building of the institute was a collaboration between Jonas Salk, a scientist, a medical doctor, vaccinologist as he would call himself, in collaboration with the great architect, Louis Kahn. And this collaboration was to create the crucible in which foundational scientific discoveries would improve human health. So the way that they designed the building was one that has an open floor plan. You can go from one side of the building to the other without running into a dividing wall that's permanent. And Jonas Salk anticipated by many years what Frans Johansson described in the Medici effect, the idea that the best ideas are born at the intersection of disciplines. So when you look at the work that's done at the Salk Institute, you can see that the 52 faculty that we currently have are studying every important discipline in science that is important for not only improving the health of people, but also of improving the health of the planet. For example, through our Harnessing Plants initiative, which is meant to mitigate the effects of climate change by improving the ability of plants to capture carbon. A fascinating program. And of course, our cancer biology and neurobiology programs that will attack two of the most important problems that have to do with an aging population. So we hope that we will follow Jonas Salk's admonition and advice that we must always do work where we realize that our greatest responsibility is to be wise ancestors. That is, to think that what we are doing now, how it will have an impact on generations to come. So with that, I want to tell you about how I came to this point of being a scientist. Now, I grew up in a middle to lower middle class neighborhood in Los Angeles in the San Fernando Valley, not the good part of the San Fernando Valley. And my dad was a furniture salesman and my mom was a stay-at-home mom raising 2 children. And my parents told me that I could go to any college that I could get into as long as I could live at home and pay my own way, and that it should be in Los Angeles. And so that pretty much committed me to going to UCLA, which is a great school. And I had a wonderful education there. And at UCLA, I had to work my way through college. I worked in a lab for 2 professors, both of whom had come from Harvard. And it was through their mentoring that they gave me the confidence that I could go from being a kid growing up in an average home with no scientific background. I could go from UCLA to one of the best institutions in the world, Harvard University. That's where I went to do my graduate training. But my graduate mentor decided that it would be better for him to work somewhere other than Harvard. So he went to Salt Lake City, Utah, where I joined him as a graduate student in my 3rd year. And I finished my training there and then went on to do postdoctoral studies at Stanford University. So from my modest roots in the San Fernando Valley, I went to a great college, to a terrific university, a set of terrific universities, and then ended up at the Salk Institute, where I've been my entire career since 1979. Now, one of the really great things about being a scientist is that you get to talk about your science to scientists who are located throughout the world. And so this picture shows some of the many places that I've been, but I think one of the most exciting places that I got to go to was Stockholm, Sweden in 2007, when my mentor, Dr. Mario Capecchi, received the Nobel Prize in Physiology and Medicine for figuring out how to genetically engineer mice. If you want to read a fantastic, fascinating, empowering story about how you can go literally from being a street orphan to winning the Nobel Prize, read the story of Mario Capecchi, because that's his story. Now, I want to turn to the next topic about what is science. People have different ways of thinking about it. I think about it as just a system for acquiring knowledge, where we use observation and experimentation to describe and explain natural phenomena that surround us, whether it be, how plants grow the way they do, why we kill plants by giving them too much water, why plants avoid shade. Why is it that people develop cancer? And how is it that we can treat cancers to give people better and longer lives? So through this system of acquiring knowledge, you learn how to formulate testable hypotheses. This is at the basis of the best science. And the best hypotheses are those that immediately suggest the experiments to test them. You'll understand this as I get into what kind of work we're doing in our lab. First issue is, how do you decide which of the many possible questions you could imagine answering, being empowered by this scientific method, to spend your time on? Now, for me, the answer was pretty straightforward, because both of my parents died of cancer. That's what motivated me to spend my time studying cancer. And I want to give you the example of breast cancer. It's one of the cancers we study in the lab, although the methods that we use could be used to study any cancer, or in fact, any disease. Again, the power of science is it gives you ways of asking questions in a way that they can be answered. So why is it that we decided to study on breast cancer? Well, I think there are two important issues. One has to do with relevance, and the other has to do with feasibility. As you can see here by the statistics, in the United States, breast cancer is a worldwide problem. Let's just talk about the United States. About 1 in 8 women, more than 10% of all women in the United States, will develop breast cancer during their lifetime. This amounts to about 270 or 300,000 women a year developing breast cancer. And it's not just the disease related that's present in women. About 1% of the cases will also be found in men. This results in about 40 to 45,000 deaths per year just from this one kind of cancer. Well, in order to understand cancer and how to study it and assess this issue of feasibility, we have to understand the organ in which the cancer arises. The other thing that I wanted to bring up has to do with these statistics called 5-year survival numbers. And the important point here is that if a person is diagnosed with cancer in their early stages, breast cancer in their early stages, 99% or more will be able to survive at least 5 years. And this is if the cancer has stayed in the breast, it hasn't gone to lymph nodes, it hasn't spread to other parts of the body. But if the cancer has gone outside of the breast, gone to the lymph node and spread to other parts of the body, then the 5-year survival plummets to 28%. And this movement of the cancer cells from their site of origin to distant parts of the body is called metastasis. So this problem of metastasis is what we really have to be addressing in order to improve lives of people who have been diagnosed with cancer. Now, the mammary gland is actually a pretty simple organ. It's designed to make milk, and there are just 2 types of cells that comprise the mammary gland. There are these outer cells that are shown in red here called myoepithelial cells. They're muscle-like cells, and they're designed to squeeze out the milk that is produced and secreted into this hollow space that's called the lumen. Now, the cells that make the milk are shown in green here. They're called luminal cells. So you'd think, well, breast cancer must be pretty simple. There are only 2 kinds of cells in the breast that can generate cancer, these basal cells or the luminal cells. That can't be too hard to study. Well, turns out the problem is more complicated than that. And because the problem is so complicated, you might recall that before President Obama left office, he empowered then Vice President Biden, now President Biden, with developing what was called the Cancer Moonshot. And this was an idea to get the best scientists in the world to work together in order to attack all of the many diseases that we lump together under this single name cancer. And the reason he empowered President Biden to do that is because President Biden lost his oldest son, Beau Biden, to glioblastoma, a form of brain cancer, one of the cancers that we work on here at the Salk Institute. Okay, so here's this cancer moonshot. President Biden now has a lot of money in order to do that. Are we going to be able to do it? And is the moon shot the right metaphor? And I don't think it is, because there's an elephant in the moon. Now, you know, to get to the moon was one thing. It's a planetary object. We know its trajectory. We know how far away it is, and we can use relatively simple math in order to program a rocket in order to get there. And we've done that a number of times. Cancer is much more different, is much different than the problem of getting to a known object at a fixed place in the universe. And I'll explain to you why. The problem has to do with this issue of tumor heterogeneity. So I told you that there are only 2 kinds of cells in the breast. Well, it turns out that if we look at individuals with breast cancer, we can identify many different kinds of breast cancer. And in part, they can be defined by the kinds of genes that drive the growth of the cells that produce those cancers. Now, we don't have to go into these various names. The importance of defining the cancers by the genes that are relevant to their growth is because that can suggest the kind of drugs that we can use to treat them most effectively. So those cancers that are driven by activation of these hormone receptors, we can use drugs that antagonize the activity of those hormone receptors, so-called anti-estrogens or aromatase inhibitors. These are compounds that inhibit the production of estrogens. Well, there are other kinds of cancer that may not have these genes driving them. They may have another kind of gene. It's called HER2. It's a gene that's a growth factor receptor gene. Well, we have many drugs that can inhibit the function of this growth factor receptor HER2. We can use those drugs to fight this kind of cancer called HER2 positive breast cancer. But then there's a significant number of breast cancers, about 20% of the total, that are highly aggressive. They often form metastatic lesions, those kinds that really decrease overall survival. These are called triple negative cancers. And the reason for that is because they don't have any of these hormone receptors and they don't have HER2, so they're negative for all of these. And the only way that we can treat these cancers currently is using generally acting agents that we call chemotherapy. Now, these drugs can be effective for some period of time, but often people relapse with very dangerous disease. And also giving chemotherapy is associated often with many kinds of side effects that are difficult for patients to tolerate. So we need to understand, we need to be able to give these triple negative cancers a name so that we can fight them with drugs that are much more better tolerated by the patient. Now, there's another problem, and that has to do with the heterogeneity of cells, differences of cells within the cancer itself. Remember, I told you about this kind of cancer that's driven by the HER2 protein. If we take 3 different sections of a single patient's cancer that was called HER2 positive, we can see that only this section in the middle is one that has cells that actually have elevated levels of the HER2 protein indicated by this kind of brownish reddish color. So what that would mean is that if we gave these people a drug that antagonizes HER2 called Herceptin, only the cells in this middle part would be sensitive to it. The cells in other parts of the cancer that don't have expression of this gene HER2 would be resistant. So it gives you the idea of cancers being composed of different kinds of cells. Some of the cells will be sensitive to the drugs that we give them, like Herceptin. Once Herceptin does its job and eliminates those cells, new cells grow. So it's a problem of whack-a-mole. You get rid of some, other ones come back. So we really have to understand this issue of how is it that cancers develop the different kinds of cells that they contain? How is it that intratumoral heterogeneity arises? Because it's this intratumoral heterogeneity that creates an imprecision for even the most precise medicines that we currently have available. This gets to this issue of formulating hypotheses that suggest the ways, the experiments to investigate them. And I'm not going to go into what the surgeon Francisco Durante said in 1874 about how cancers arise and how even cells of different types arise within the cancer. Basically, his idea was that cancers can be driven by inflammation. And if that inflammation lasts for a long period of time, it can cause the cells within the cancer to change their look, kind of like how a chameleon changes its color. And we now call that process cell state reprogramming. And he further said that these cells could change the way they behave to the way that would resemble that found in the most primitive cells of the organ that was affected by the cancer. In other words, the cells of an adult would reprogram to an embryonic state. In the case of the breast cancer cells might reprogram to the cells found in the embryonic mammary gland. And these cells are very plastic. They're able to change because that's part of their normal developmental program. So he reasoned this could generate this intratumoral heterogeneity that he was able to observe just using a microscope. So his hypothesis suggests the following way of looking at breast cancer. We could just go through, in this case, looking at mice, which we can study, and looking at different stages of the development of the mammary gland, starting in the embryo, going just past birth, and looking into the adult, and isolate cells from each stage of development, isolate individual cells and determine what genes they express, and then ask, do cancers express genes that also relate to any of these stages of development? If Durante's hypothesis is right, then at least some of the breast cancers or some of the cells in breast cancers would resemble those by their transcriptional programs that are present in these early embryonic stages. So we did that experiment, and lo and behold, what we found is that these triple negative breast cancers, the kind that the pathologists have called the least differentiated, sure enough, resemble the cells that we found to be present at early stages of embryonic development, the cells that are most plastic and able to change in response to the environments in which they're placed. Now, then the next question becomes, well, what do we do with this information? Well, two postdocs in the lab at that time, Chris Dravis and Jay Young, decided, well, cancer is a problem of the expression of genes, and specifically the way that the chromatin, the DNA, and how it's packaged to express genes actually expresses genes that are relevant to the cancer. So they looked for genes that were specifically expressed in the embryo, that were also expressed in these basal-like breast cancers. And they found a gene called SOX10, which we knew to be a master regulator of cell state due to the work of others. And what they also found was that this master regulator is itself regulated by wound factors. That was interesting to us because we knew through the work of pathologists such as Hal Dvorak that cancer looks like an ever-healing wound that never heals. And what Chris and Jay and Nikki Lytle found was that when we exposed cells, either cancerous or normal, to these wound factors, and if they expressed high levels of SOX10, as they do in many of these basal-like breast cancers, the cells begin to move. They acquire this ability to move out of their normal environment to distant environments. That's the process of metastasis. And this answers the so what question. So what if we find that SOX10 causes this state change where the mammary cells change their state to become motile? Well, it turns out SOX10 is a master regulator. It controls the expression of other genes. We looked for other genes that it expresses, and we found that some of them encode proteins, say this one, KIT, which encodes a tyrosine kinase. This is the kind of protein that Tony Hunter has studied for so many years, that these kinds of genes are often found to be overexpressed or mutated in cancers, and many drugs have been developed against them. Turns out a drug has been developed against KIT, and it has been used therapeutically, although not for breast cancer. And now we're studying these drugs for new purposes, their ability to intercept metastatic disease. So by this foundational research that we've done, looking at cancer as a reflection of normal development and influenced by inflammation and wound healing factors, we found new molecular drivers for metastatic disease. We are developing new targets for the interception of metastatic disease. And we even think that we can use this knowledge to apply to the new field of immune therapy to harness our immune systems to fight against these dangerous cancers. So my take-home lessons for today are, anybody can be a scientist if you're naturally curious and you want to know how the natural world works. The scientific method teaches us how to ask questions in answerable ways, and it's applicable to every discipline. Just because an idea is old, such as Durante's, doesn't mean that it has no merit. In fact, nobody had the ability to study that question until recently. And now we find that it has great relevance to not only breast, but many other cancers as well. And if a hypothesis is testable, important, and will advance knowledge to improve human health, then we are obligated to test it, to be the wise ancestors that Jonas Salk told us to be. And finally, although I've talked to you about breast cancer, we found that many other cancers also reprise these embryonic programs to lead to this big problem, the elephant in the moon, that is intratumoral heterogeneity. And now we're learning how to deal with it, intercept it, and improve the lives of people diagnosed with many forms of cancer. Now, this kind of work isn't done by me, certainly, not even by a few people, but by many people. We have lab meetings. We've had them during COVID. This is how we had one lab meeting during COVID. Everybody masking, socially distanced. This is our mascot, a wallaby, wall labby. And this is the way we usually celebrate on the patio at Salk, that we hope that you'll visit at some point and see it in its true glory. And of course, we couldn't do this without adequate funding. Thank you very much for your attention. I hope you have a great time visiting the Salk this week.
Thank you so much, Dr. Wall, for sharing with us your research and your passion for science. We do have time for a student question. So our first question was submitted by a freshman, and they would like to know, what do you think was your best learning experience?
You know, I have to tell you that my best learning experience was my first failure. Science is often met with a lot of failure. Mother Nature is very smart. And what that failure taught me was to figure out whether my hypothesis was wrong, and urged me to think about new ways to test it, or whether I had done the experiment wrong and the hypothesis was right. And that first failure taught me what I was really good at. And I hope that I've been able to transmit these abilities to understand whether the hypothesis is wrong or whether your approach is wrong, and we need to try new approaches to address important questions.
Excellent. I love that you highlight how important it is in science to recognize failure and really see it as an opportunity for growth rather than an opportunity to give up. So for students that have tuned in, thank you so much for joining us. Now you're going to have the opportunity to get to interact with some of the Wall-labbies, as Jeff had mentioned. We're excited to welcome you into the lab for a virtual lab tour coming shortly. Thank you so much, everyone. See you soon. Welcome back, students. We are welcoming back Dr. Jeff Wall. We got some amazing questions after the keynote presentation, and we wanted an opportunity to be able to answer a couple more. So thank you so much, Dr. Wall, for coming back with us. So we have an opportunity to do a little bit more student interactions.
That's great.
So the first question, so the first question that we received has its two parts. Dr. Wall, what led you to start to explore and further your knowledge on breast and pancreatic cancers? And as a follow-up to your presentation, will individualized therapies mean that we need to come up with many more medicines?
Those are two really good questions. And there are a few different answers for each one. So what got us interested in breast cancer was, one, as I mentioned in the talk, how common it is. About 1 in 8 women in their lifetime can have experience a diagnosis of breast cancer. And as I also brought up in the lecture, when you study cancer, you have to think about the model that you're using. How accessible is it? What kind of experiments can you do? And the mammary gland, the breast, is a non-essential organ. I mean, it's essential for aesthetics, obviously, in humans, and for producing milk for the next generation. But if a woman is diagnosed with breast cancer early, for example, you could do a mastectomy and the woman can live a perfectly normal life. So in that sense, it's not an essential organ. So we can do many genetic manipulations in experimental animals that enable us to study environment interactions in this non-essential other organs that are essential. The other thing is that the mammary gland is a superficial organ, so there are many ways that we can study it. You might not believe this, but we can actually build windows into an experimental animal so we can actually observe the mammary gland developing and cancer developing. Also, we can take out the mammary gland and we can study it in the laboratory, and we can take out its cells and we can study them in the laboratory. And importantly, we can even take the stem cells of the mammary gland and transplant them back in and get a whole new mammary gland to develop. I mean, it's really amazing if you could see this. If you have the mammary gland stem cells that are labeled with a green fluorescent protein and you put them back into an animal, that animal will develop a green fluorescent mammary gland that'll produce milk. The milk will be white or normally colored, won't be fluorescent. But that to me is just totally amazing and proves that the cell that you isolated was actually the stem cell of the organ. You can't do that for many other organs. Mm-hmm. So there are many experimental reasons for studying breast cancer and because of its medical relevance. Pancreas cancer goes to a different issue, and that is how very difficult it is to diagnose and to treat, and how commonly it turns lethal and kills people. And as I tried to indicate in the lecture, you have to study those projects that really interest you. Mm-hmm. And this problem of why isn't pancreas cancer susceptible to treatments that other cancers seem to be sensitive to, or why can't you use immune therapy against it, are important and challenging questions to address and answer. And I used to be president of this organization called the American Association for Cancer Research, and suggested at the time that I was president that they develop basically what we call now a crowdsourcing mechanism for raising money for cancer research. That became Stand Up to Cancer, which is the nation's first nationwide telethon to raise money for cancer. And as part of their first fundraising efforts, they raised money for pancreatic cancer and formed what was called the Pancreatic Cancer Dream Team. And I was fortunate enough to be the part of the SALT team that became part of that Stand Up to Cancer Dream Team. And it's gone on now, and it has several clinical trials using strategies that were developed here at Salk. So we hope that those will enable more effective treatments of cancer, of pancreatic cancer in the future. So those are some of the reasons that I've gone in to study those two cancers.
And so will the need for individualized treatments, will that require us to create more medicines? You talked a little bit about some of the clinical trials. So will we need to create more medicines if we try and pursue this individualized medicine approach?
The answer to that is yes and no. Yes, because we're learning how individual the cancers are between people. And as I pointed out in the lecture, when cancers grow, they develop different kinds of cells. So a treatment that's good at one time might not be good at another time. The more we learn about cancer, the better able we will be to prescribe therapies that'll be targeted most effectively against an individual's cancer. Now, does that mean that that cancer will be completely different from another person's cancer of the same type? And I think what we're learning now is that we may actually need fewer drugs than we initially thought of. I'll give you one example. So there was a drug that was developed to work on chronic myelogenous leukemia, CML. CML used to be a death sentence. There were no treatments available for it. Then lab scientists and medical scientists found out that there is a particular gene that is activated. It's activated by two chromosomes coming together in an inappropriate way in something called the chromosome translocation. And that creates an abnormal gene. And that gene encodes a protein. That protein is an enzyme called the tyrosine kinase. Tony Hunter at the Salk Institute, trying to understand how viruses cause cancer, discovered tyrosine kinases as a new kind of enzyme in animal cells decades ago. And it turns out now that those proteins, these tyrosine kinases, are found to drive many different kinds of cancers, and drugs have been developed to inhibit their activity. Getting back to CML, turns out that a drug was developed for a completely different kind of gene, a different cancer driven by a different tyrosine kinase, but that kinase is related to the one that's activated in chronic myelogenous leukemia. The drug is called Gleevec. So here's a case where a drug developed against one cancer actually didn't work in that cancer. It worked in another cancer driven by a related protein. And now you fast forward another 10 or 15 years, and there's another cancer completely unrelated to chronic myelogenous leukemia, and it's called GIST, which is a abbreviation for gastrointestinal stromal tumor. It is activated by a tyrosine kinase, different from the one in CML, but in that same family that's inhibited by Gleevec. In fact, the enzyme that drives GIST is the target that Gleevec was initially designed to inhibit. And now Gleevec is used to treat GIST as well as CML. So here are two completely different cancers. Cancer's a genetic disease. The gene that tends to drive both of these cancers that are unrelated is of the same class, not the same gene, but the same class, and the same class of inhibitor can be used on both of them. I can tell you about many other examples of that. For example, in breast cancer, there's one subtype I told you about, the HER2 positive one. And there are drugs that are targeted against the HER2 protein. One of them is called Herceptin. There are other cancers that are driven by HER2. That drug can be used for them. And the story is continuing to evolve. So on the one hand, we have to know the genes that go awry that drive a person's cancer so we can tailor the therapy to them. Those genes will be different in different people, but on the other hand, the classes may be the same, so we can use the same class of drugs to treat those unrelated cancers.
So it sounds like there could be opportunities that when we learn more about cancer on a genetic basis, we might be able to use medicines that already exist, but just have a new application that could be successful. So the following questions kind of align.
So that's very good. So that's called drug repurposing. And in fact, there are a lot of studies that are ongoing now to look at drugs that have failed in the past that might work now or in the future, because we better know the targets that are relevant. So good point, Madison. Thank you.
Yeah, absolutely. So this next question kind of aligns with treatments, but it talks a little bit more about pancreatic cancer. How come cancer treatments that have worked for other cancers do not work for pancreatic cancer? And what causes the pancreatic tumors to be able to block immune cells?
Yeah, so actually, those are two really good questions, and the reason for treatment failure is partly related to that second point that was brought up about what caused the pancreas cancer to block immune cells. So I think that there are a few reasons. Let's talk about why pancreas cancer is lethal. First, it's generally diagnosed at late stages. So a person doesn't present with any symptoms. When they do, often the cancer is metastatic. So that's the second part. Pancreatic cancer tends to metastasize very easily, and it's close to the liver, and it metastasizes to the liver, and that's a very dangerous situation. Another reason is that as the pancreas cancer evolves, it changes its microenvironment. the constellation of other cells that grow with the cancer. And the cancer educates the microenvironment then educates the cancer, and they form this self-reinforcing cycle of growth. And one of the consequences of that growth is that the cancer develops what you might think of as a hard outer shell, like a nut. It's called the desmoplastic reaction. Fibroblasts in the cancer microenvironment deposit a bunch of collagen. That collagen gets cross-linked and it gets really tight. In fact, sometimes when the surgeons go in to take out the pancreas cancer, they can actually bend their instruments because the surface of the cancer is so tough. And so you can imagine that with that kind of tough structure, the vasculature becomes enfeebled. It's called a hypovascular cancer. And so there aren't a lot of blood vessels going to it. That can prevent a lot of drugs from getting to the cancer. Okay, so you've got the shell. The shell prevents the immune system from getting in. It also prevents drugs from getting in. And finally, because the cancer is hypovascular, a lot of parts of it are hypoxic. And the response of the cells to that hypoxic environment is to produce gene programs that cause them to become resistant to a bunch of drugs that they would normally be sensitive to. So there are all these reasons. It's the biology, the ecosystem of this abnormal organ that is now the pancreas cancer that has defeated our attempts so far. So one of the approaches of the Salk team in this pancreatic cancer dream team has been to normalize this stroma, to treat the cancer from the outside in, to then be able to treat it from the inside out. So to create vulnerabilities that enable the immune system to work better, that enable chemotherapies to work better, and that still limit the amount of toxicity that the patient will suffer.
Yeah, it sounds like it's a culminating of all these factors kind of make it this like super villain of cancers that make it exceptionally challenging to treat and to research as well.
Right, absolutely true. Although we have now, I think some of the students visited with Danny Engel's lab, and she studies organoids, and you can actually create or mimic kind of a pancreas cancer in the laboratory environment. You can create these organoids. So you take a chunk of the cancer out, you grow it under specific conditions in the laboratory, and they form these three-dimensional structures that kind of resemble the cancer from which they came. And we're trying to do other related systems where we actually take slices of the tumor and grow it in special dishes where the cancer can get oxygen from below and fed from above. And in that way, and we can look at these growing cancers using a microscope. So in that way, we can study them over time as well as in 3 dimensions. So there are new technologies being brought to bear on this very important problem. And a lot of them are being implemented or developed here at the Salk Institute.
So changing gears a little bit, you talked about this in the beginning about breast cancer being very common in your talk. So why is it so common?
You know, I think that there are, again, there's never just one reason in cancer. There's often many reasons. And some of them are pretty trivial when you think about them. Again, the mammary gland is an external organ, and there are ways that people can be taught to feel the breast in such ways that they can tell whether there's something abnormal going on. So self-diagnosis turns out to be one way that people can go to a doctor early on and say, gee, there's something wrong here, could you check it out? And then a cancer will be found. There's also mammograms that can detect cancers. That's another reason. Third reason is that the development of cancer can be linked, in breast cancer, can be linked to certain metabolic conditions. So obesity is a risk factor for the development of breast cancer. Since there's an epidemic of obesity, that links to an increase in breast cancer. And, the mammary gland, in order to produce enough cells to produce the amount of milk that's required to feed a baby, is going through at each cycle, these periods of growth and retrenchment associated with the the periodic cycles of women, as well as when women get pregnant. And so you get increases in the number of cells, and the cells are exposed to different hormonal environments that are conducive to growth. And growth, of course, cancer is a genetic disease. And as cells divide more, they have a greater chance of developing mutations. And so the more you have a chance for a cell to divide, the greater the chances that it'll acquire a mutation that will enable it to divide further to generate what we then diagnose as a malignancy. And the other thing is that, of course, you can't ever think about the cancer as just the growth of the cancerous cell itself, but rather the environment in which it grows, just like I was just talking about pancreas cancer. So in response to obesity and other conditions, the age at which a person has their first period, for example, the microenvironment can change. And those microenvironments can change in ways that enable cells that have acquired a potentially cancer-causing mutation to manifest itself in terms of cell growth. So just because a cell has acquired a mutation doesn't mean that it will grow. It needs to be in the right environment to grow. And in fact, studies have been done years ago using animal models that showed that the growth of a cancerous cell could be suppressed by putting it in a normal microenvironment. So that tells you how powerful the environment is for restraining cancer growth. I'll give you another example. If mutations were just enough to allow cancers to grow, then you would expect that those areas of your skin that were always exposed to the sun has ultraviolet rays that cause mutations, those areas would have all kinds of the cells exposed to the sun consistently would have lots of mutations. You should get lots of cancer in that area. Well, it turns out this is the area that's most exposed to the sun, right above, right in your eyelid area. You don't put sunscreens here because the sunscreen will get in your eye. So this is always exposed to the sun if you're out in the sun. So people have actually taken biopsies from this region and they've sequenced the DNA in the cells in that region. They have all kinds of mutations. But the structure of the skin there remains normal and is one contributing factor to suppressing cancer growth. So I've given you a number of reasons for why breast cancer is common. But the next question is, well, are other cancers common and we just don't detect them? And the answer to that is yes. And here's a reason that I know the answer is yes. Turns out a study was done a long time ago in men who were in their late 70s and 80s who had died in car crashes, and they had no other known cancer history. And people at autopsy, pathologists asked, did they have any evidence of cancer in their body? And it turned out that men aged 80 and above, I think it was, I'm probably wrong on the exact number here, but it was upwards of 90% of them had benign prostate cancer. So they were going to die with their cancer, not from their cancer. The cancer was contained. It was contained in the prostate capsule. So only when the cancer gets out and metastasizes, as it happens in the breast, when it metastasizes to the bone or the brain or the lungs or the liver, does it become life-threatening. If it stays there in the breast, within a duct, you'll have cancer, but it's not going to kill you. So cancer is much more common than we think. If you diagnose it, you count it. If you don't diagnose it, it doesn't mean it wasn't there. So our immune systems are typically doing a great job of keeping it at bay, as is the normal structure of our body.
Awesome. Thank you so much. I think you're right. I think it's hard to pinpoint a single reason, and we're not aware. We can't be counting problems that we're not aware of. So this one's talking kind of pivots a little bit back towards the research, and maybe you can share your insight. But can RNAi be used in cancer research? If you could share anything you know about that topic.
Sure. So RNAi, or interfering RNA, sometimes called shRNA or short hairpin RNA, was discovered many years ago in Basic Research Lab. In fact, I think initially the initial evidence of it came from plants, and then Hal Weintraub started to use it in his cancer research, and in studies of normal cell developmental control, because cancer's just development gone awry, and Hal Weintraub was interested in normal development as well as cancer. So siRNA is a short molecule of RNA that's complementary to a normal strand of RNA that you would have in your cell. And when it pairs with that normal RNA, the cell recognizes this as abnormal and it degrades that RNA. So imagine that you had an RNA molecule that encoded an abnormal protein that led to cancer. So you could imagine using this magic bullet of the siRNA to put into the cell to get rid of that cancer-causing gene product. So the question was, can it be used in cancer research? And the answer is, it's used every day in cancer research. But if you really meant, can it be used in cancer treatment? That's a different question. And that gets to the issue of, how are you going to deliver it to the cancers? Are you going to put it in a pill? Are you going to put it in a virus? If you're going to put it in a virus, is the virus going to get incorporated into the cell? If you're going to put it in a virus, how do you make sure it's not going to get incorporated into a normal cell where it could do some damage? If you're going to put it into a pill, how do you make sure it's going to get into every one of those cells within the cancer? Because if it doesn't, then the cells that don't get it will just continue to grow. So it's been very challenging to deliver molecules like this to cells. People have used nanoparticles and a variety of other strategies. And so far, it hasn't been very successful. But, you know, there may be other ways of using it other than trying to get rid of cancer-causing genes that would make it successful. And one of the ways that, now I'm not just thinking about siRNA, I'm thinking now about RNA-mediated approaches. So let's think about the COVID vaccine made by Moderna or Pfizer. There, what you have is you have an RNA molecule that's packaged inside a nanoparticle that is delivered into your muscle. But it actually isn't, its effects aren't coming from it getting into the muscle cells. Its effects are coming from those nanoparticles getting into dendritic cells and other resident cells of the immune system that are present in the muscle. And when the RNA is expressed, it makes part of the spike protein. And that part of the spike protein that it makes initiates an immune response, which creates a memory. So imagine using RNA approaches as vaccines for cancer. So that is one approach that is being tried now by BioNTech, which is the collaborator with Pfizer to make the Pfizer vaccine, and a variety of other companies. So I think when you think about how you're going to drug cancer, you really have to think about this problem of how you're going to deliver the drug to the cancer, and can you really deliver it effectively enough to have the widespread effect that you're going to need. So yeah, siRNA is used every day in cancer research, not so much in cancer treatment yet. But maybe those with imagination will enable it to be used in the future.
The power of the word yet. I love that. It's potential for the future. All right, it looks like we have one final question. So that is going to be, what are some of your favorite topics to teach and why?
You know, what I really like to teach about in cancer is just how adaptive the cells are and how they become so adaptive, how they're able to meet virtually any challenge that we throw at them, and how this reflects some of the basic processes that cells go through in normal development. And to understand these links between normal development and cancer and how it goes awry. I think that's really a fascinating. There's a lot of interesting history that comes into that, even from the mid-1800s, as I presented in my lecture with Durante, where he didn't have very sophisticated tools, but he just observed what the cells look like, to the present day, where we're able to isolate these cells and determine every single gene that they're expressing, and we're still puzzled by how they do what they do. So that to me is really fascinating. But honestly, I like to teach what people are excited about. And so in my lab, we research what excites people the most. You don't want to study something that's boring everybody. Otherwise, it's just hard to come in. Rather, science is kind of hard. Mother Nature is very, very clever. And usually we think we have a great idea only to be proven wrong. So the real trick is to study what you love to do, so that in the face of being shown that you're wrong, you'll have the courage to go in another day and figure out another approach. And I
think that was, yeah, I love that we're ending on following your passion. It's something we obviously can see that you do, and it's apparent from all those that in your lab that we interacted with. So thank you so much for taking the time to answer some more student questions. We're really excited for students that are watching this. If you want to learn more about the Wall Labs research, we'll definitely include a link to this video so that way you can check out some more information. Thank you so much for watching. We'll see you next time.
Thank you,
Bye.
Madison. Thank you all the students for coming to High School Science Week at Salk. Thanks for the great questions.
Thank you.